Hi all, I'm Angela Di Capua, a postdoctoral researcher in Vaisakhi Research Group in Chemistry and Biochemistry, and today I'm presenting one of my projects conducted in collaboration with Gopalan Lab in the same department. Many of you may be familiar with Salmonella infection. Salmonella is a foodborne pathogen that causes gastrointestinal problems. This bacterium makes its way to the intestine when the affected person eats raw or uncooked meat, eggs, or egg products. In the United States, there are nearly 1.45 million cases of Salmonella infection every year. During infection, a molecule called fructose asparagine is used as a nutrient by Salmonella. This small molecule is metabolized through a catabolic pathway that involves different proteins, encoded by a cluster of genes called FRA. These genes can be turned on or off by a protein using a locking key mechanism, with the entire cluster controlled by a master switch. In our research, we want to understand the mechanism related to the FRA cluster and the lock protein. In my work, we use a mass spectrometry, an analytical technique that can determine the mass of small molecules as well as large biomolecules, such as protein and protein DNA complexes. In particular, with native mass spectrometry, we can investigate in gas phase protein and protein DNA complexes in their native-like state, while preserving their interaction. We use mass spectrometry in combination with other biochemical techniques to determine if FRA, the lock protein, is able to bind DNA, and also to identify the key that can unlock the protein and allow the protein synthesis. Using native mass spectrometry, we have been able to confirm that FRA is a dimeric protein, 2 copies together, and that it binds the DNA. We also found that 6-phosphofructose aspartate, an intermediate of fructose asparagine metabolism, is able to bind FRA and works as a key that leads the protein to no longer bind the DNA. So 6-phosphofructose aspartate unlocks the FRA protein. The protein can then release the DNA, which will start the transcriptional activity. The use of native mass spectrometry, together with other biochemical techniques, provide an important contribution in understanding how a key pathogen utilizes a nutrient found in the animal gut. This works as implication for drug discovery.